All right, good afternoon, everybody. It's a pleasure to introduce our next speaker, which will be Josephine. Josie is a faculty member in the Department of Internal Medicine in the Division of Digestive and Liver Diseases. And like Ralph, I remember doing this in 2008, so it's really a wonderful forum for our new faculty to get to know the community and for us to get to know them. Josie came to started her undergraduate studies at UC Berkeley. She's originally from the West Coast. Then moved to the middle of the country to Northwestern for her medical degrees, and then completed the migration to the East Coast for internal medicine training at NYU. And she became chief resident there. After internal medicine, she wants to go into GI, and I remember Josie coming down to UT Southwestern for interview for fellowship. We lost the battle to Penn, where she went and trained under Gary Wu, who's a well-known gastroenterologist who studies host microbiome interactions, and also worked with Mark Goulian, who's a microbiologist, studies sort of the microbe side of that interaction. So there's a microbiology theme here. Josie also has a secondary appointment in that department. She had a very interesting paper in Science Translational Medicine describing the role of amino acid metabolism in the relationship between the host and the microbiome during inflammatory bowel disease, which is something that she does clinically. We were delighted to recruit her in the late part of 2021. She comes already with a Barrows Welcome Award for career in medical sciences. And she's been here a little over a year and already has had tremendous success working with Kim Orth. They got this tremendous HHMI award for emerging pathogens. So it's a pleasure to introduce Josie, and we look forward to hearing what you have to share with us.
Hi, everyone. I always have to move the mic shorter because I'm short. So I'm so excited to speak here to you guys today. My first family, internal medicine. There are so many great faces here. And my second family, microbiology, is also here in force. So I feel like I'm already amongst friends, which is awesome. Today, I'm going to talk to you a little bit about bacterial biofilms. So, the human GI tract, which I'm fascinated with, of course, as both a gastroenterologist and a microbiologist, is home to the densest population of bacteria within the human body. But, how these bacteria actually exist within the GI tract and, how they form communities has been really a subject of a lot of debate. So, what are biofilms and can they really exist within the GI tract? Well, bacterial biofilms can be considered to be a bacterial lifestyle. They consist of kind of colonies of bacteria that are surrounded by some sort of bacterially produced materials. And they tend to be a source of cooperation, resistance, and a way to survive for various types of bacteria in nature. But the human intestinal tract has long been thought to be a site where biofilms are probably not going to be very prevalent. And the reason is because within the colon, there's goblet cells that are, embedded within the epithelial cell layer, and they secrete a lot of mucus. This mucus kind of contains immunoglobulins, which will keep bacteria from penetration of this particular mucus layer. And it's also the site of a lot of constant mucus and cell turnover over time. Mucus kind of grows at hundreds of microns per hour, and then epithelial cells can be shed at the rate of billions of cells per hour in the small intestine and about 10 times slower than that within the colon. So add to that kind of constant peristalsis, which means food and water is constantly moving through your GI tract, and it makes the time scale of biofilm formation, which is thought to take days, at least hours, to be a little bit less plausible. That said, we've often seen these biofilms that are associated with other disease, GI diseases such as inflammatory bowel disease, a disease that I treat within clinic, and also colorectal cancer. In all of these kind of studies, they've been able to stain large amounts of bacteria within very close proximity to the human epithelial cell layer. But I found this to be really interesting because even though we've kind of touted intestinal biofilms as a source of disease, pathogenesis, we don't really know what an intestinal biofilm really is. And we don't really know if there's really any good in vivo models to study an intestinal biofilm. And all of these studies, furthermore, have concentrated on pathogenic conditions and diseases. So are there even biofilms in a healthy intestinal tract? And does that really help in terms of bacterial proliferation in a commensal kind of environment? Well, I love E. coli for various reasons, and I think it's a great model organism. It's a great model organism because it's a commensal. It's associated with dysbiosis, which means a change in the microbiota in response to any kind of disease. It can be what we call a pathobiont, which means that it doesn't necessarily cause disease in certain situations, but can cause disease in other situations. It's also very genetically manipulatable, and it is able to colonize the mouse intestinal tract in long-term experiments. So, interestingly, we had noticed a little bit earlier that, I was interested in nitrogen metabolism in the intestinal tract. And when you knock out all the two component signaling systems, which are these kind of key mediators of bacterial signal transduction, there's only one particular knockout that gave you a colonization advantage in mice, and that was NTRC. It's a two-component signaling system that senses low nitrogen conditions within the intestinal tract and then activates a number of downstream transcription factors for response. And what we realize is that we can knock out NTRC as a particular gene and put this particular strain into mice to confer a fitness advantage in vivo. So the way that we do that in my laboratory is we pre-treat mice with antibiotics and polyethylene glycol. So we kind of give them a colonoscopy prep as well. And then we inoculate them with, bacterial strains in competition over the course of 5 days. These mice then kind of have a lower activation energy for changing their intestinal microbiota. And then we're able to collect the stool from these mice and quantify these fluorescently tagged strains. And what's really unique about this particular system is we don't have to use germ-free mice in these microbiota use-based studies. And two, I'm able to induce stable colonization for here I show about a month, but we can really induce stable colonization for about 3+ months with just one single course of treatment. So we can actually see how bacterial these microbiota colonies, communities actually evolve over time. So we can see here that both in kind of conventional notobiotic environments, if you knock out NTRC, you're able to grow these particular bacteria as opposed to wild type with like a 2 to 3 log growth, a competitive advantage, even though in vivo on the left, they don't do quite as well. So for some weird reason, the mouse intestinal environment is a really great competitive environment for these NTRC mutants. Because we fluorescently tag these mice when we sack them, we can also see where this competitive advantage is happening, both kind of radially in the mucus and the lumen, and also longitudinally throughout the small intestinal tract and the large intestine. So we were really interested because we saw this competitive advantage mainly in the mucus layer, the intestinal tract, and we were thinking, well, can we really tie this to a particular phenotype? And we discovered that this particular strain, despite having this fitness disadvantage from grown in a planktonic state, is able to form better biofilms in vitro assays. So we started using this as a model for intestinal biofilm formation. And I think that we can use this and harness this particular model to understand both the structure of an intestinal biofilm, explore the crosstalk between the host environment and biofilm formation, and most importantly, because I am a physician scientist, I'm interested in exploring the interaction between bacterial biofilms and intestinal diseases. So one proof of concept, that experiment that we've done is to kind of systematically knock out different extracellular matrix components to intestinal biofilms. Curly is actually the major extracellular matrix protein that's produced by Enterobacteriaceae. So this was our first target because even though it's important in biofilm formation, its role in intestinal tract biofilm formation hasn't been well characterized. We were able to make this mutant that doesn't form any curly. It doesn't basically form intestinal biofilms, as you can see on the left, or biofilms, but it grows very similarly to its parent strain. When we inoculate these strains into mice, we can see that it loses the competitive advantage that's seen in the NTRC strain, our good biofilm former. So the blue circle dots basically show that they are no longer able to compete as well as wild-type E. coli. When we give them this kind of gene operon back, it's able to restore this competitive advantage. So you can see that the blue circles and the black triangles are kind of acting, they're growing the same way on the right graph. We then are continuing to explore many of these other extracellular matrix components as well. Secondly, we can use this particular model to explore the crosstalk between the host environment and biofilm, particularly looking at small molecules that the host wants and the bacteria want. So one of these proof of concept experiments has to do with iron, which has been shown to be a very desired micronutrient, which is fought over by the host, which secretes iron chelating molecules, as well as bacterial pathogens. We first showed that using a competitive iron chelator, we're able to inhibit biofilm formation in our mutant strain at a particular concentration that doesn't actually affect growth. And so we were able to, again, reproduce a genetic knockout model of what we saw through the competitive inhibitor. And we showed that when we knock out this particular strain's ability to chelate iron, it no longer can outcompete its wild-type strain in the mouse intestinal tract. When we were able to give back this particular operon, it's able to, again, have that competitive advantage, which was seen before. But most importantly, we really, really care about how bacterial biofilms are important to intestinal diseases. So we've been exploring whether or not these biofilms are important in mouse models of colitis, and whether or not we can use commensal strains to outcompete pathogenic strains to make a difference in disease. One of the models that we've been working with is the SAMP model of colitis. These are kind of immune-mediated models of colitis in which these mice, like inflammatory bowel disease patients, develop spontaneous colitis in their small intestines. So what we do is before they grow to the age where they develop spontaneous inflammation, we clean them out, inoculate them with our bacteria, and then we again are quantifying these particular strains. In this case, we're trying to outcompete a particularly cholitogenic strain of E. coli, NRG-A57C, which has been shown to cause worsening disease and inflammation. And you can see here that when we co-inoculate these mice with wild-type E. coli and this NRG-A57CR pathogen, it really doesn't do anything. But when we add in our NTRC mutant, we're able to decrease pathogen load by a number of logs. When we kind of look at the colonization of these particular strains in these mice, we see that the numbers of wild type versus NTRC, Delta NTRC E. coli are actually not different. So there's actually similar amounts of both types of E. coli within these intestinal tracts, but where they're located, I think actually really matters. So at the end of the day, using this model, we were able to show that, we're able to probe both molecular basis of biofilm formation, kind of how the host interacts with bacteria via small molecules and competitive in a competitive way. And most of all, we were able to show that in vivo models of disease and also in health, we're able to affect colonization. I wanted to really thank the community that's made this possible. I still have a lot of collaborators, both at Penn and also newly here at UT Southwestern. And my laboratory has really grown with the addition of graduate students, a postdoc, a scientist, and an instructor, but always welcome new people who are interested in both basic and translational research. Thank you so much.
All right. Great. Do we have any questions?
Hey, great talk, Josie. So, yeah, nice talk. So I wonder, what is the biological nature of the colonization fitness? Like, survival, proliferation, or something else? And does it matter for the modeling of pathogenesis?
Yeah, so in terms of whether or not, so thank you, Eugen, for your question. Eugen is part of my GI family. And so basically, and you're asking me, how does this biofilm formation actually allow this particular strain to outcompete pathogens, and how does that actually happen? Well, it's interesting. We kind of look at both engraftment and long-term colonization. And what we can see is that even very early in the time course, we're starting, when you first kind of gavage these strains in competition with each other, in the very beginning, we're putting in equal amounts of both types of bacteria. So within the first few days, you don't really see kind of a change. But within the first week, you start to see some differences, and that gap in colonization, or really engraftment, widens. So I think that is already happening within the mucosal layer very early on. And then later on, at about the 2 to 3 week mark, when I think that more stable colonization happens, we actually see a further widening of that gap, and then that kind of stays constant. So I think that our commensal strain is able to outcompete the pathogen, both in the engraftment phase and also in the colonization phase, long-term colonization. Now, whether that's due to kind of it being able to penetrate the mucus layer a little bit earlier is something that we're actively investigating. So as you know, we recently got our own confocal microscope within our laboratory, and that's something that we're really, really working on visualizing through immunofluorescent staining. Thank you.
All right, great. We're going to have to move on to stay on time, but let's thank Josie again for a great talk.